Okay, so we are talking about proteins. That's what chapter 4.5 was about. Make sure you keep taking your notes. It was a fairly long reading assignment, maybe not as long as the one before it, but the good news is you only have 2 more reading assignments from here, and one of them is shorter, and then the other one is really short, quick, and easy. And we will have the test Friday of next week on September the 11th, so make sure you're getting prepared for that.
Football game.
There's a football game that day? Darlington?
Yes, our first one.
Really? Home?
Yes.
A home game?
How are you going to make us have a test?
Oh, well, you want to rip that Band-Aid off. It'll be great. Get to take this test, have a lot of fun taking the test, and then you might be able to get a chance to watch the football game. Okay, so there are quite a few slides in this, but with a number of them, I don't have a whole lot to say except just to introduce you to the concept. One thing with all of these chemicals we've been talking about is that there is a unit in the future where we will spend weeks focused on just a few of these chemicals and how they interact with cells and how the cells use them in different ways. So I'm trying to keep it, I don't want to say basic, I mean some of this stuff can be a little complicated, but I'm trying not to delve into all the depths of all these topics because we will see them come up again. These first 2 slides, we're going to look at each individually briefly in terms of the different functions that they're broken down into. Okay? And again, all of these are from your book, so if you have access to your book, you can look at them, so there's no need to draw anything. But as you can start getting a picture of in these first 2 slides, proteins have a lot of different functions in the cells. They are a very, very important and diverse group of chemicals. So in a minute, we're going to talk about how they are used as enzymes, how they're used by the immune system, how they're used to store important smaller chemicals like amino acids, how they're used to transport materials, how they're used to send chemical signals, how they're used to send other types of signals across small spaces between cells, how they're used in movement, and in general construction of the living form. So there's 8 different functions that we're going to look at each individually here, and we're going to start with one of the many important ones, enzymes. So we're already down, we got 3 slides already. Look how fast we're moving. Okay, enzyme. That is a word you've probably heard before, and it's because there are many, many very important enzymes that keep you alive literally from 1 second to the next. All right, cells rely on enzymes. Living things rely on these enzymes. Now, they fancy it up a little bit here with enzymatic proteins. Basically, an enzyme is a protein. You should be familiar with what it does. Does anybody know what an enzyme does? What's the purpose of an enzyme?
Speeds up reactions.
Speeds up reactions. If you aren't familiar with that concept, that's what you want to know. An enzyme speeds up reactions. It's a lot like in chemistry, if you talked about a catalyst, that's essentially what an enzyme is. It's a catalyst, but it's a protein. It is a protein. All right? So, there are a lot of reactions that happen in the human body. Almost all of them can be divided into reactions that build things and reactions that break things down. So you're either building up or tearing down. All right? You have to do both in your day-to-day living functions. Now, this is a very simple example, but I think it is a great way to look at enzymes. in this case, we see kind of a general chemical reaction that is occurring where some sort of larger molecule is being broken into smaller subunits. So this might be a disaccharide, two sugars that are bonded together. All right? There's a lot of disaccharides that you eat every day. Your body will typically want to take those disaccharides, those slightly more complex sugars, and break them down into individual monosaccharides. There are enzymes that do that. And essentially what happens with an enzyme is there is some sort of geometric location that a chemical will bond to. The enzyme can then act on that chemical and help make some sort of chemical reaction occur at a much faster rate. So in this case, once this more complex molecule bonds to this location, the bonds between it can be broken, and you have successfully broken the chemical down. This might still happen without the enzyme, but it might take years instead of seconds. They can really speed up this process, and that's critical. One thing you probably want to note, and I believe your book discusses this, is that location, which is kind of shown here as this shape that these other chemicals can fit into. This location is known as an active site. An active site. That is a term that will be used throughout the year. An active site. And essentially, the chemical fits into that active site, fits into that location, and that's when the enzyme can do its work. So what you find is the shape of these enzymes is important. And we're going to get to that and talk more about that at the very end, about shape. Geometric shape. There is another term before I move on that I want to mention, and that is a general term that we use for whatever it is that the enzyme is bonding to. And your book will use this term a lot, so you want to be familiar with it. But that term is substrate. A substrate. Substrate. So we'll say that the substrate interacts with the enzyme, and the enzyme acts on the substrate. At what location? The active site. Very good. So that's the kind of vocabulary, that's the kind of terminology you want to be able to understand. We'll talk about a lot of enzymes this year. So knowing what in general they are at this point is important. Okay. Some of these others I won't spend that much time talking about. More or less just referencing certain words to be familiar with. So they talk about defensive proteins. This is a word that I want you to be familiar with, and in this day and age, you probably are. The term antibody. Antibodies are proteins. And you use them to fight infections, whether it's a bacterium or a virus. Now, how exactly they work, how your body produces them, what triggers all of that, that is not something we're looking at right now. But I do want you to be familiar with the fact that antibodies are another example of proteins. And if you ever have a virus or a bacterium, your body will work very hard to create antibodies, which look like these kind of Y-shaped structures, that will attack specifically that invader. So when you get sick, your body tries to learn about what's making or causing that illness, if it is some sort of tiny critter like a virus or a bacterium. And then it will flood your bloodstream with these antibodies, and the antibodies basically just swarm around whatever that germ is and take it out. Try to kill it that way by essentially smothering it and making it incapable of continuing the infection. Antibodies. Defense. Proteins are also used for storage. You do not need to know any of these specific examples. Just be aware of this as being one of their functions. They give examples like the protein in milk, the protein of egg white, the protein in seeds. Anytime you eat something like peanuts or walnuts, if you're not allergic to nuts, a big reason people enjoy them is actually because of the protein content. They're a nice, filling, hearty meal or snack. A lot of times these proteins, which all proteins are made of amino acids, but these proteins exist so that you can chop up the protein into its amino acids, take those amino acids, and then build whatever proteins you need to build. That's usually what you're doing. And we'll talk about amino acids here as well in a minute. All right, another function is to transport materials in and out of cells or around the body. So we talk about transport proteins. Again, no specific example, like they mentioned hemoglobin here. No specific example needs to be memorized. All right? There are actually a lot of ways that this transport can happen, and one way, a very common way that we will talk about a lot in the future, are what we call transport proteins. These transport proteins. Now, we haven't talked about cells yet, but you may be somewhat familiar with what cells are from another class. Every cell is going to have some kind of outer layer, typically called a cell membrane. And in that membrane, you have these transport proteins just all throughout it. And essentially, they act like doors. You can open them and close them. When you open them, things can go in or out of the cell. When you close them, it prevents that from happening. Again, we will talk about many examples of this. But here's the $50 question. Let's see how much y'all have absorbed here in this class so far. What do we see right there? What is that? Little... Yeah! It's a phospholipid. That's right. That just made me very happy. Oh, and they have cell membrane listed here, so at least it's marked. Yeah, there's the hydrophilic head of the phospholipid where the phosphate group is, and then the fatty acid tails. Very good. Very good. We will talk about a diagram like this much more coming up. Other functions. Some hormones are proteins. Others are actually lipids. Don't write that down. So not all hormones are proteins, but some hormones are proteins. And one that you're probably familiar with, but I will not expect you to memorize because we're not going to talk about it specifically really much this year, but insulin. You've probably all heard of insulin in relation to what disorder? Diabetes. Basically, insulin, which is a protein, it's not an enzyme, it's not storing anything, it's not helping transport anything itself directly. It's just a signal. And that's what hormones are. They are chemical signals that your body sends out in the bloodstream. They travel somewhere else and tell other parts of your body to do things. So when you eat a meal, the sugars from that meal tend to get into your bloodstream from your digestive tract. That typically triggers a release of insulin from your pancreas. And that signal of insulin being in your blood tells other cells to take sugar out of the blood, and then your blood sugar level starts to drop. So in the case of diabetes, some people can't make insulin, and other people, the insulin just doesn't successfully send the message. They make insulin, they secrete it, but their cells and their other parts of their body don't actually react to it. But another example of how proteins are used by the body as certain hormones. Similarly, they can be involved in kind of a more direct communication between two cells in the forms of receptors. This is actually the unit we end this semester with. We essentially spend an entire unit talking about how there are little proteins on one cell that can detect chemicals, and then they tell that cell to do something or to not do something. Your nerve cells rely on this a lot, which is the example they give here. But you'll have one nerve cell send some chemicals out. The next one will use proteins to essentially read those chemicals, and then the signal can continue down that nerve cell. It's kind of a horrendous unit. I hate teaching it. It's my least favorite unit of the year. I don't hate teaching it. I love teaching. It's just not as fun. Welcome, or goodbye. All right. One you're probably a little more familiar with, even though it might not sound that way from the use of these terms up here, but contractile proteins are very important for animals because, as you can see, these are the proteins that make up your muscle tissue. And from that term contractile, you get the root word of contract, which you can also call flex. And it's possible to contract and relax your muscles because of these interactions between different types of proteins. Again, words like actin and myosin, I am not going to ask about. I'm not asking about specific examples until we get on into the year and a specific example comes up that is important. But proteins can work together and even by themselves sometimes to not just contract. So this interaction can actually allow itself to kind of pull in together, meaning contract, or to kind of release that tension and lengthen out, which is to relax a muscle. But there are also other proteins that you don't see a whole lot in humans, but they're called well, actually you do see a lot of them in humans, I think about it, but not with your eyes, they're all microscopic. But you have these motor proteins that can actually kind of turn and move in different ways. Not necessarily contracting, but there are movement generating proteins that exist. And those tend to be called motor proteins. But in general, you can actually use these to move parts of the body. Again, just like I mentioned how like peanuts. People enjoy eating peanuts. Some people, I love peanuts. They're delicious, especially roasted and lightly salted. What a great treat. People like eating peanuts because of the proteins they have. Well, muscle is meat, okay? So anytime you eat hamburgers or steaks or a chicken wing, that is simply the muscle from that animal. And because muscle is so reliant, or the purpose of muscle is to contract and relax, and those processes are so reliant on protein, when you eat meat, when you eat the muscle from another animal, you are getting that protein, and that is a nutrient that your brain rewards you for eating. That's why people like hamburgers and hot dogs. Even though hamburgers and hot dogs probably contain a little bit more than just muscle. Other little bits and pieces of the animal get crushed up in them. And then finally, and then we're going to move into what exactly a protein is. These general structural proteins are important, that just to hold things together. You have things called connective tissue in your body that just keeps your bones together, your muscles attached to your bones, your skin positioned properly on your face. Collagen is an example of one of these. It's not for movement, it's not for storage, it's not an enzyme. It's simply there as like tension wires would be for a bridge, just a structural component. When you get older, and this won't happen to you for a while, but it sure comes fast in life. Some of these proteins start to break down and they don't really hold your skin to your face or to the rest of your body as well. And what happens? Yeah, that's what wrinkling is. That's right. Your skin starts to sag and pull away a little bit from where it's supposed to be because there aren't these structural fibers made of proteins there to help. Okay, so that's what proteins do. Again, no specific examples need to be memorized. Just understand that they have all these different possible functions. Now, what are proteins made of? They are made of amino acids. These are 20 amino acids, pretty much the complete list of amino acids that you use to build all the proteins you need to build. And I know it might be a little bit hard to see because all 20 of them are fit on here at the same time. But I want to really point out what's in purple on each one. These purple boxes in this diagram are all exactly the same for every amino acid. And I'm going to draw something because they do something I don't really like here. They put it kind of in this ionic form, which is okay. But I'm going to, just off to the side, draw make sure this is in my video here. Essentially, you have this kind of core chain of a nitrogen and 2 carbons. So this is where we start seeing other elements come into play. Every amino acid is going to have this nitrogen atom here. Now, how many bonds can nitrogen make? Nitrogen can make...
4, no, that's carbon.
Not quite 4, that's carbon, good. But it's 3. So one of those bonds is bonded to the next carbon, and then you have 2 hydrogens bonded to it. Okay? This central carbon always has a hydrogen, and we're going to come back to it. And then in an amino acid, at the end, you always have a double bond to an oxygen and then a hydroxyl group. Now, the diagram here specifically, as much as I like this diagram, it drops off this hydrogen and it adds a hydrogen over here, and it has kind of a negative positive charge, and that's fine when they're by themselves, but that never really comes into play for us. Okay? Now, the reason I draw this is because this is the same for every amino acid. This is kind of the core to every amino acid. But then right here there's a bond, and a lot of times you will see the letter R written here. And there's no element that has the symbol R. And if you've looked at this diagram, you probably have an idea of what the R represents, because the R here is a variable. And we actually called this part of every amino acid a variable group. And what this R represents is that it's at this point on an amino acid that you could have any number of these different possible groupings of atoms. So that's a diagram I would definitely kind of note, and if you can't see it that well, it'll be on the video. I will not ask you to draw this from memory, so don't worry about that. But sometimes on a test or even a quiz, I might put this structure down as a question and just have you identify it in a multiple choice way as either being a carbohydrate, lipid, component of a protein, or component of a nucleic acid, which we'll talk about next class. And you just need to know that, or recognize that this is an amino acid which is part of a protein. So again, here is where the diversity and versatility of proteins kind of arises. It's this location where you can have all these different, what we call side chains. Now, it might just be like in the case of glycine, this is an amino acid where at that point, you just have one hydrogen atom. How simple. But there are some with kind of more complex atomic rings, and even some, like cysteine here, that has the element sulfur. And this is actually when we talk about Chopins, and we had sulfur as a sixth element that you find in all living things. It's because of amino acids like this that you find in almost all living things. Sulfur. What does sulfur smell like?
Rotten egg.
Yeah, sulfur is kind of stinky. It's kind of stinky. You find sulfur in fireworks, so the fireworks smoke has that kind of like stink to it, that smell to it. Rotten eggs smell like rotten eggs because a lot of the egg is what?
Sulfur.
It's proteins made of amino acids, some of which have sulfur. That's right. And in fact, that's roadkill. Anything stinky that you can, well, not anything stinky, but a lot of stinky things in the world are stinky because of sulfur, typically found in amino acids which make up proteins. So these are the monomers. Now they're all, again, the same in this one location, this core part of the molecule, but they do come in different forms. The last thing I want to say, and we'll revisit this, is that because of these different forms, and they kind of group them here like this, some of these are hydrophobic, meaning they don't mix well with water. Some of them are hydrophilic, so they actually have a kind of water-attracting component. Some have positive or negative charges, which they indicate as being kind of an acidic or basic-y kind of chemical. So there's a lot of chemical diversity here. All right. So to get a protein, you need dozens of amino acids at the very least. It's something around 50 amino acids that you would need before you could actually have what we call a protein. And they all have to be bonded together. Does this look familiar?
Yes.
It's a dehydration reaction. That's right. This is what we do to get monosaccharides to stick together into polysaccharides. It's what we do to get fatty acids to stick to a glycerol molecule to make a molecule of fat. And it's what we have to do to get two amino acids to bond together. So when we pull out the hydrogen here and the hydroxyl group on the other amino acid, we are producing water, and that leaves open the bonding locations for these two amino acids to covalently bond. Now you may remember from last class, when two sugars are covalently bonded, we call that a glycosidic linkage. And when we have this covalent bond holding the fat molecule components together, we called it an ester linkage. Here, and I do want you to know this term, peptide bond is the name we give for the covalent bond. It's a covalent bond like any other, but it's the covalent bond that actually holds the two amino acids together in the chain of amino acids you're building. Be familiar with this term peptide bond. You see it from time to time. Now, as you start forming these peptide bonds, by bonding these amino acids together, you begin to make a long chain of amino acids. But it might not be a protein yet. But we have a word for chains of amino acids. We use the prefix meaning many, which is what? Poly. And then we stick that in front of this term peptide. So we call it a polypeptide. You will see that term, polypeptide, and it just means many amino acids. I guess it was too weird to call it a polyamino acid. So they used the term polypeptide. But what we're doing right now as we build these chains of amino acids is we are getting towards a protein. So on this next slide, I want you to see kind of how the entire protein sometimes can look or be represented. These can get really, really, really complex. You don't need to know the difference between these models. And all I'm doing here is just showing you how this is a very complex molecule. Here we see every atom represented as kind of like a sphere. And you can see that there are tons of these atoms all packed together. Now they have other ways of showing protein structure because, I mean, what a mess. That's a mess. What does that tell you? Well, you can use what they call ribbon modeling, where they kind of show these, looks like ribbon to give you a general idea of the shape, or a wireframe model where instead of a wider view, you get kind of a thinner view of the polypeptide chains. But again, the purpose of this is to show you the complexity of these molecules. And I want you to keep in mind, while it looks like a total mess, if these were enzymes, there is some location here that is considered to be an active site. Even though the shape is complex, the shape is terribly important. And here they even like highlight, here's a molecule, a substrate molecule, and the protein, even though it looks wildly chaotic, has a very intentional three-dimensional piece to this molecule that the target molecule can fit with. There are millions of proteins, billions. I have no idea how many. You have 20 different amino acids you can fit together in any way you want in chains of hundreds or thousands of amino acids in a row. The possibilities become astronomical. So getting to this final shape is what actually makes a protein a protein. If it doesn't have a shape that is useful for one of the functions that proteins serve, then it's just a polypeptide. It's just a bunch of amino acids. But if it has the shape that gives it its function, then we can start talking about using the term protein. So, kind of in the final stretch here, I want to talk to you about these 4 terms. We have primary, secondary, tertiary, and quaternary structure. And they all have pretty simple definitions, even though it might not look like it from some of these diagrams. But every protein you could talk about in kind of 4 ways. 1, the primary structure has a simple definition. It's the order of amino acids. And that's it. So on the most fundamental level, when you talk about a protein, the most fundamental thing to know to start out with is which amino acid is the first one, which amino acid is the second one, which amino acid is the third one, all the way through the dozens or hundreds or thousands of amino acids. Now, instead of writing out their full name, like instead of writing out glycine, every amino acid is given kind of this 3-letter code. You do not need to know any of them, but if you ever see something like this, I do want you to think in your head, that is an amino acid and not a sugar or a fat. So these 3-letter abbreviations are very commonly used for amino acids. So primary structure is simply order of amino acids. That's it. That's what I want you to know. Secondly is the secondary structure. And again, while it looks complicated and they use fancy complicated names, it's actually got a simple definition if you break it down. But based on the order, based on that primary structure, these chains can twist or fold. Okay? So here we see a twisting that occurs, and here we kind of see this zigzag folding that can occur. So secondary structure, you're simply talking about twists and folds. That's it. Twists and folds. Now, unfortunately, scientists don't like using fun, simple terms like twist and fold. So they call the twists alpha helix. Do you know what the plural of a helix is if you have many of these? It's a weird pluralization. That's very close. Helices. You leave out the X and you put like a CES in, I think is how you spell it. So you have these alpha helices, or a twisting is considered an alpha helix. Helix means to spiral. And then the folds they refer to as like beta pleats or beta pleated sheets or a beta strand. I mean, there's a reason for the complexity, but sometimes it feels on this level a little much. But the AP exam loves to throw out terms like alpha helix and beta pleated sheet at you, and all they're talking about is how these amino acid chains aren't straight like the ruler is here, but they can actually be twisted around. twisted around a helix, or just folded into these sheets. But that's the secondary structure. Based on the primary structure, based on the order of amino acids, that part of the chain may twist or fold. Twist or fold. Now, that's not the extent of the shape. You have a third level, which that's what the word tertiary means. It means third. You have a third level where you look at a larger portion and how these twists and these kind of folded parts fit together. Okay? So this tertiary structure of a protein, the third level of complexity, is understanding how you can have many twists and many folds all on one chain. So we're not looking at one twist, we're not looking at one fold, we're looking at kind of all the twisting and folding on the entire chain. That's the idea. We're close to being done, guys. We're close to being done. And then finally, you have the fourth level of complexity, referred to as the quaternary structure, which just means fourth, quaternary. And this is where you have multiple, basically tertiary structures stuck together. This is where you can take 2 or 3 or 4 chains that have been twisted and folded, and those separate chains are bonded together or fit together in some way. And this is where you actually have what we would call a protein. But understand, all these levels are linked. The order of amino acids reflects whether there's twisting or folding. All that twisting and folding can give a chain of amino acids a certain shape, and then multiple different chains can come together to make a really complex glob, all just made of amino acid. And once you have that highest level, those chains are all together and you have the entire structure, that's what you would call a protein. Okay. 2 more slides. I'll make it quick. So here we're kind of looking at why. Why would a twist occur in a chain? Why would a fold occur in a chain? Why would different chains actually stick together to become an even more complex structure? Well, it again goes back to these side chains, where I wrote the letter R here. What is right there determines what the shape ends up looking like. And this is a really, it might again look complicated, but a pretty simple way of showing all the possibilities. So this is your polypeptide chain, or polypeptide backbone, whatever you want to call it. This is the polypeptide, meaning many amino acids. So there's an amino acid here, and here, and here, and here, and here, and here, and here, all the way along. Each one of these amino acids in this chain has some unique atomic group that's coming off of it. But what happens is they can interact with each other. So an amino acid in one part of a chain can actually interact with an amino acid somewhere way down the chain. They might form hydrogen bonds. They might form these even weaker bonds known as van der Waals forces, which is something you might have talked about in AP chemistry, but probably not in regular chemistry, I don't know. You can get sulfur atoms actually just make a covalent bond, a nice strong bond between sulfur atoms. Or some of these are actually positively and negatively charged, so they make an ionic bond. So they kind of, it's like socks out of the dryer. They can stick together in all sorts of different ways. And that's just what's happening here. This one chain can stick to other parts of the chain and cause this bending or spiraling or folding to occur. That's how you get the shape. And the shape means everything. Which takes us to this last slide here that is related to the shape of a protein. And the only word I really care that you know on this slide that you'll probably ever see is denaturation. To denature. Now, this process can go the other way. But again, this is a polypeptide chain right here. Looping around, folding around. Maybe there's a couple of chains here that were individually built and are now stuck together. It's hard to tell. It's such a freaking mess. But it's got a very specific shape. And since it has that shape, it has some function that it could perform. So it's a protein. But what happens sometimes is if you heat up a protein or expose it to, say, a strong acid or base, or maybe something else could happen, typically heat will cause the shape to kind of fall apart. And it can denature. And if a protein loses its shape, it doesn't work anymore. This is why if you have a fever over like 104, once you start getting over that mark, a lot of your proteins in your body will denature and lose their shape permanently. And when you start losing all your enzymes and all the things that hold your body together, that causes some serious damage, and that's why it can be fatal to have a very, very high body temperature or fever. Okay, that took so long. 50 minutes. You can stop the video and give me one